السلام عليكم ورحمة الله وبركاته. بسم الله والحمد لله والصلاة والسلام على رسول الله صلى الله عليه وسلم ومن والاه. رب اشرح لي صدري ويسر لي أمري واحلل عقدة من لساني يفقهوا قولي. إن شاء الله تعالى. الحمد لله رب العالمين ورب العرش الكريم. We are ahead of schedule and I'm just planning preparing for the lab today. Students are going to be focusing on plants and their vegetative morphology following the drought treatment or control that they did 2 weeks ago. And thereafter, they'll be using cross sections, making them using a microtome to observe the different tissue cell types found within the celery stalk. Inshallah ta'ala. Barakallahu fikum. Assalamu alaikum wa rahmatullahi wa barakatuh. لا إله إلا الله لا شريك له له الملك والحمد يحيي ويميت بيده الخير وهو على كل شيء قدير. Good morning. Sorry. Inshallah tomorrow. I'm gonna be able to show, or hopefully you're able to see the night. practice on top of on Ummah, not on the stalking, one has the bond, creation for it. So like the first down, let's say that we have this is the base kind of speed up, that means in time, and not the last and kind of find it and options use your in the last Um, also, we have another so obviously the issue if there is a group, I believe few members have that. So hopefully, in terms of the ratio, we have them on board for the group. So we these last week clubs. So once you're done with your last week, use the clubs, take them accordingly. You have how you can work. Just make sure that during the lab that you have those on, that you are dealing with the microphone, which is first, and also video, which can be done. And Adam's not that you know about the so just want to make sure that that your lab quiz, is your quiz on either side? Mandatory course, you have to go on to the boxes, build the module, read the questions, and you should have access at least to the private system where you have options to lab to. Make sure once you're there that you are doing the library, you need to lab background, process, but bear in mind that you're not obviously breaking academic taking. Well, just bear in mind it's 10 minutes, right? So try to do it. Yes, 10. Make sure that the drop running any water bottle or very important. So when you do the cold, also then I think they're in trouble. Just a reminder for the lab quiz, it's timed for 10 minutes. You can begin if you haven't done so already. You can use your lab manual, a calculator if needed. You need a laptop, they're at the back. And accordingly, if you um, once you're done, you can review today's exercises for the manual. Is Eva Jill here? Salma Garcia Rodan? I'm just verifying for attendance online as well. Joshua Shank? Joshua Shank? You're Joshua Shank? Oh, perfect. Okay. So let's see. I believe I have everyone in attendance. Let's see. Okay. We don't technically validate us. Let me see. He wasn't here last week, so I don't mind. Yes, I'll get it. like there's like right. No worries. Thanks, Joshua. So I think we'll wait just a moment. Just a kind reminder, everyone, if you haven't done the last quiz, if you want to as well, keep in mind that like the entirety of today's lab and future labs are on times, like quick times are allocated, so just bear in mind that we won't do that quick time. That's each time, once you're done your lab quiz and you review the manual, please don't forget to wash your hands. To also wear gloves for today's practical because we'll be using sharps. We are also taking out grass from the plants that we used 2 weeks ago. So just bear that in mind as well as the use of TBO, which we'll discuss shortly or during the exercises today. Just need to confirm if Salma Garcia is here and Eva Jill. Everything okay? Everyone's doing good? So just bear in mind that once again, make sure you do not have water bottles or teacups or phones, especially outside on your bench top without letting me know. Uh, Phones we'll talk about briefly in due time. Inshallah, let's begin. For those that don't know, my name is Mohammed Saeed and I'm a postdoc, what we could say, at CSB, Cell Systems Biology. Congrats for making it to your second week. And I'll put my email address shortly for those that want to get to know a bit more about me. But in the meantime, We have some important slides to go through, right? If you haven't seen from before, we'll be focusing on plant vegetative morphology through looking back in the past, about past 2 weeks for the FLR preparation of data analysis for shoot biomass and length, as you can see behind me. And then after this, actually, that's the investigating effects of drought on plant growth section, which we'll do first. And then thereafter, the final component will mainly focus on plant vegetative morphology using the celery cross sections you find, and that you will prepare from the stalk itself in the middle bench today. Briefly, just want to also highlight for safety, because we're dealing with sharp objects, please bear in mind that you don't touch the actual, essentially, metal piece within the microphone. That's very sharp. Right, so you'll find them on the left edge of each microphone. Also bear in mind that you're using razor blades when you're cutting and fine-tuning the sizes of your celery stalks for microphone cross-sectioning. So just bear that in mind, that you're very safe and you're keeping in mind of yourself, but also time with your group. So hopefully by now everyone has completed the lab quiz. Show me raise his hand if you have. Want to make sure. Okay, no problem. So we'll wait maybe 2-3 more minutes, if possible, and that's going to be to review what we see from application with now carrying forward. Reminders, you come into the lab, make sure that your fire, if you need to fire or anything, put them outside, it's in brush up, and then you certainly no problem for us. Honestly, I want to do all the hydration, but definitely you don't want to have fire, so then nothing. When it's lit, another, the meters essentially go out down. If you have first aid here, gloves here for today. These are your plants from 2 weeks ago. And I'm also going to be reading and this being your first week. So if you want to essentially the lab, sorry, like our knowledge. And shortly we'll discuss how you'll essentially prepare your calculation for 2 length environments. Besides that, any questions? And I also want to remind everyone that as you come in, make sure that you buttoned up your lab coat. Make sure that your hair is tied to the back if you have hair uncovered. Also, generally speaking, you want to have closed-toed shoes. So just bear that in mind. I know we're obviously wearing in the winter, but it's very important, especially when we get back to spring. Now, hopefully, Joshua, do you mind if I just continue forward or? Yeah, okay. So in the meantime, attendance has been done. Information of the FLR, formal lab report. How many of you have not taken AO1? You don't mind raising your hand. So everyone's familiar with the formal lab report, right? Perfect. So for context, just to briefly highlight some key features, it's worth 5%. It's focusing on the past 2 labs of lab 1, preparing for your plant growth assays essentially, and then now today uncovering the treasures of your results, the biomass, the shoot length, as well as the qualitative observations you have in comparing the drought treatments with your control. And we'll be doing these different components as a lab together for collecting the data. But it's important that you bear in mind that the deadline for it is on the 30th. Sorry, it'll be posted likely, yeah, I guess this coming Friday, but the deadline is March 3rd. I highly recommend before our next lab to start working on your introduction. Very, very simple, and even the methods if you can, depending on the criterion for the formal lab report. But essentially, the introduction can even be done in advance pretty much based on what you have from last week as your intro and from the methods as well. But then your results is pretty much what we'll be focusing on today. I also make mention of a few points if there's time for it. But briefly, the scientific method. By the way, can everyone see the slides? Down by that side. Miriam, can you see the slides easily? And Faizan, you can see from that end. Want to make sure everyone can see, which is important for accessibility. And in terms of really the scientific method, I've touched upon this briefly, but I try to take a more holistic approach, being able to understand and appreciate the different concepts that you see in the sciences and beyond them. So just bear in mind that when you essentially observe for the purposes of your alphalar and more broadly in STEM and beyond, try to think about how you can essentially form hypotheses, which we'll be focusing on today for your research hypothesis, but also more broadly how you could be able to create your own experiments, be it for future labs, right, B-level courses, or even hopefully down the road for your own lab, right, and trying to uncover new pieces of technology, new modes of software, new modes of methodology, and even uncovering new findings in different organisms and how they could perhaps relate to human health. And that's the mode of investigation. Communication is where you present those results in literature or presentations, exploration for future analysis, which is often mentioned in your discussion. Now, in a controlled experiment, there are different factors and questions you have to bear in mind and to take into account. So amongst them, you could see like being very specific in terms of details, like what species are you using? Are you using Polyana Arabidopsis? Are you using like musculus, which is like a mouse, a house mouse, versus like essentially a long flowering plant. And there's so many other species, right, you can think about. But on top of that, right, having species is important and fundamental for your research question and your hypothesis. Following that, you want to talk about your sample size when you're focusing on your methods. Are you looking at only like 10 cells? Are you looking at 10 different plants? Are you looking at 100 different organisms, etc.? And if you have replicates, that also bolster or augment the potential for your statistical analysis and also to create greater and more reliable findings. And then you have the independent variable, your dependent variable. Independent one is the one that you're using to measure, right? So one that you can manipulate indeed. In our case, right, we were focusing on 3 different treatments of drought in 2 conditions, treatment 1 and 2, and you had our control, which also leads to the following point. But dependent, yes, and the following point being actually what we'll talk about in a few slides, I think. Yeah. Essentially the values that you control, right? So it could be the amount of water you've added as the entire bench or as the entire lab, right? We added 50 mils in total of water for each treatment. Also controlling for the amount of light. As you can see on that side, but also on that side, the amount of light that's usually added is maintained so that way you don't have to run the risk of any unexpected findings occurring because of uncontrolled variables that weren't accounted for. So those are things that you want to bear in mind for also your equipment, right, in terms of different errors that may exist, but in terms of also influencing the values that you get for your dependent variable. And that is the variable that it can be measured actually as well, rather than manipulated as an independent variable. So dependent variables like shoot length and biomass, which we'll be measuring today, are key traits for you to be able to, discern different patterns and different parameters that you will focus on in your hypotheses. Which brings me to the next slide, and that is your statement, i.e. research hypothesis, and that is where you're focusing on testing a given relationship in your experiments. There are 3 main conditions. They don't always have to be, I say that's more abruptly, that sense is really that you want to be very specific, of course, very detailed, right? As I mentioned earlier, the different conditions, the different species, the variables that you're accounting for and the controls as well down the road in your methods. And then being able to test those different relationships and with your variables is also needed. And perhaps that it could be proven false, right? We have like the null hypothesis, alternative hypothesis, and based on your statistical analysis, you should be able to verify whether or not your results either support or reject your alternative hypothesis, and conversely, if you accept or reject your null hypothesis. Now, this is a sample for an upcoming lab of what your research hypothesis could be like, right? And so bear this in mind because you want to be articulate and as specific as possible. Yeah, you can see my mouse, but let me just highlight this for you all. On the left-hand side, for this side of the bench, you have temperature, right? That's something that you are going to be analyzing, right, as your independent variable. You're changing it up and down with daphnia, or which is like water fleet. And you can already see it specified here. Okay. But then you have the effect of heart rate, right? So that's your dependent variable, right? So you see as you increase or decrease temperature, what the effect has on heart rate, physiological parameter indeed, likely what you on heart two. And then your units, which you also want to verify, right? Beats per minute as you count down the number of beats that you see on the water fleet through your microscope every so often per minute. Now, four key traits have been highlighted in this research hypothesis as your dependent variable. Actually, I'll say in order, your independent variable, temperature, your dependent variable, and essentially having an effect as heart rate, the units for your dependent variable, i.e. how you'll be measuring it in your results, and then lastly, your species name. So bear in mind that you also use the common name like water fleas here, and Daphnia, SPP, which is like species for short, if you don't know the name. But essentially use both. And you should note, this is important, Daphnia or Daphnia is in italics. So bear that in mind. Your common name is in normal script, but Daphnia is in italics. So bear that in mind. Any questions? Now, any questions? A common question actually is about stating your prediction, because often it's confused that, okay, I have a research hypothesis, maybe that's also my prediction. In reality, you actually are making an if-then statement, i.e. if your hypothesis was correct, then this is what you predict. And that's what you see highlighted in yellow as your initial hypothesis, and then bearing in mind what you predict in blue, right? So if, once again, temperature has an effect on heart rate measured in beats per minute in water fleas, Daphnia SPP, metalysis, then heart rate will be different, faster or slower, and you would specify more directly in cold or hot water than in ambient water temperature, which is ambient refers to like room or normal temperature of water. So that's something that you want to bear in mind. And once again, actually, importantly, you're focusing on your experimental parameters, right? Your treatment groups or your parameters, much less on your control, because your control is what you're going to be focusing in your methods, and that's what you'll specify there. Yes. It will be separate. It will be a separate like section, or like you could specify it after your hypothesis, but it will embed essentially what you concluded. So essentially, think of it like this. You'll mention, let's say, your FLR, both slides of your research question, or sorry, your research hypothesis, like this. And then afterwards, you would mention your if-then statement, i.e. your prediction, which is both combined. You can also simplify that if my hypothesis is true, then I would expect X, Y, Z. But for, let's say, your lab quiz, where you have to specify specifically, okay, this is my research hypothesis, this is my research prediction, that's where you actually want to specify both as directly as possible. That answer your question? Any other questions? Okay. So let's move ahead. We have 2 hours and a half. And this is your research question, pretty much of what we've done 2 weeks ago. If you look behind you, you should be able to see your bench tops flowers, or sorry, your plants, winter wheat, right? From 2 weeks ago, and the results of their growth. Just a reminder, bench A is on my left, bench B is on my right, and on this most ahead of me essentially is the control treatments, right? And then the middle row is essentially treatment one, and then the last row is treatment two. So just bear that in mind. If you move spots, that's also okay. Use the results that you have now. And actually, at the end of today's lab, we'll all be accumulating them on my computer or on the desktop here. And I'll be essentially or likely the lab technicians will upload them on Quarkus, so you don't have to worry about missing data. Any questions? No? Okay. And if you have questions, feel free to raise your hands. Just as a reminder, the details of treatments, we've talked about this before 2 weeks ago, but just to reiterate, the control did not have like essentially drought simulated, right? It was watered every day. Typically, what we'd expect is the 50 mils as we did before, or if it's specified otherwise in the lab manual, you'd go with what is shown. The drought treatment, however, was watered every other day, right? So you water one day, the lab technicians watered after the following day, right? So if it was Monday, you watered it, they would water it on Wednesday, and then Friday, and then Sunday, etc. And in the case of drought treatment two, it'd be like you watered on Monday, and then the next treatment or of water would be on Thursday, and then 3 days after that, which would be Sunday. So just bear that in mind. And of course, accounting for other variables like the light cycle, light intensity, the distance even of the light. And you can mention many other variables that essentially you would assume to be controlled and identical between containers. Now, these are questions that we saw in the previous few slides. I just want you to think about this, especially for your FLR, right? Because you want to specify these details for your methods, and likely when you come to your discussion, the different constraints that you would have had to have measured or accounted for in your controls. Now, in terms of observing your plants, we'll be doing this shortly. Just bear in mind, now is the time, actually, as groups, you can have one of your members to essentially pick up the containers that you have there and then start observing them, making notes in your lab manual. There is a section right after where you mentioned table, I believe, 2.4 for your results to actually make note of the qualitative data. So we're moving essentially to exercise 3 and then afterwards to exercise 2, so just bear that in mind. The qualitative observations are on page 45. So they're after the quantitative data that you would be inputting in. So just bear that in mind, everyone. But now, once your group or once you've found the page, now nonetheless, you can just start taking those containers now in your groups and remain seated after that. Any other questions? Any questions at all? Okay. You can have one member of your group grab your container. Make sure that you grab your essentially container, right? Your group's container. So now let's spend about 5-ish minutes just collecting the data, what you see. There's a list of questions. So just hopefully everyone's paying attention. Group 2A, just want to make sure you're paying attention. Very important questions have been asked in your lab manual as well. So just bear in mind that you answer them. They are shown on page 41. So try to make note of answering them in your qualitative observations for page 45. So as your group, just try to begin collecting that data. In about 2 minutes time, we'll all talk about them as groups and just share your results so that way others can make note of them for each replicate and for each of the following other conditions. So just look at them. I'll make mention of some, like for example, the color, the withering, right? If your plants look healthy, even generally speaking, if they look taller than other controls or other conditions you see across your bench. And also in terms of like the spotting or I guess essentially the patterns of growth that you see of the leaves or shoots of your plants. So just bear that in mind of winter wheat, Triticum aestivium. So just bear that in mind, everyone, and then we'll move to the quantitative results shortly. So has everyone been able to observe their plants? Make note of them on page 45. And it's okay to write down, like write what you observe. Don't like fake it or, make mention of things that you actually don't see, right? Just write down what you observe and make sure as well that you're paying attention, especially when instructions are being provided. Once your group is ready, once you've written down all of the observations, you could start actually getting ready to dissect your plants. And they're mentioned in the lab manual, but also in the PowerPoint slide. But I just want to familiarize with you all that you will be using this data sheet to actually input your results. So once it's time, once you've actually collected the data for shoot length and the biomass of your shoots, we'll be able to account them all. Now, I'm just going to show you in terms of a difference from your lab manual, each group is going to be making 40 counts. And there's a reason. Actually, sorry, each condition separated 20 and 20 would make up to 40 in total. I'll be separating this to make it easier for everyone. So essentially the bench, each bench condition will have 20. And then when replicated for both benches, I make it 40 in total. So don't worry about anything else beyond that. What I do want to highlight though is, oh, not that. Let's go back. The slide. Hmm. Okay. Yes, so now you may be wondering, so after you've taken those results, what other conditions what other conditions exist and what their observations for each group would be. So what I would just say is essentially from control treatment A and B, what are your observations, treatment B? How healthy are your plants looking? Are they healthy? Okay. Can you say a bit louder so that way everyone else can hear? Can you hear as well? Yeah. Okay. Continue. Okay. Yeah, that's good. Great job. Control treatment B. How is it working for you guys? Does it look well? Yeah. In terms of observations, that's great. Is the size long, thick? Yeah. Okay. Now, treatment 1A, how does it look for you guys? Okay, okay, that's okay. Okay, and treatment 1B, how's it for you guys? Okay.
Carbon is primarily it's like the foster. So that means that a couple of start besides that.
That's really good to note. And I just want to highlight for everyone, it's also good to look at the range of your entire assay. So have a look at the entirety of the container. Do you see shoots all being consistent in size, shape, or color? If not, then that's something to highlight. Perhaps that you had seeds that were more clustered in the center during the first lab. So just bear that in mind as one of the conditions. Now, treatment 2A, how's it looking for you guys?
It's pretty green, but there's not a lot of it, and it's very short and like thin.
Okay. May I ask, did you add 10 mils of seeds during the first week?
Yeah.
Yeah. Okay, perfect. Okay, so that's something to note, right? Bearing in mind that you added in the right amount, as you said, you did. So maybe this is something to think about in terms of the success of growth, right? Not all seeds perhaps have withered, or sorry, have developed and grown with seedlings and shoots. So just bear that in mind, the level of maturity as well. Now, lastly, treatment 2B, how does your plant look? How's your container look? Okay. Okay, so there's a bit of yellowing, and you observe just one more time the center. How does it look? Okay, center is also yellow. So just bear that in mind in your observations, everyone, right? You just want to always keep note of these details when you're making reference to them in your FLR. Now, for both qualitative and quantitative data. Now, the next step for quantitative observations is dissecting your plant in two ways. One is that you could cut it into thirds, or you could pick out 20 shoots with their seeds. So bear that in mind. You're taking out the seedling. So as shown here, you're taking out for exercise 3. I want to make sure you can see as well. Hopefully you can see Faisal. Okay. So essentially you're taking out the entirety, 20 of them in total, 7 on the left, right? That can be randomly selected or dissected, and then 6 in the middle and 7 on the right. So in total that makes 20. So that's the role of your group. And what I would recommend, because the idea is that after this, once and you have all of them, you can all measure them in millimeters, right? Just to know the units. But once you've taken them out with their seedlings, you measure them as essentially taking them, shoving them to at least reach the essentially active, the longest active, if you will. You can essentially just measure it as it is. Usually they're all relatively straight, but if you find them briefly, then that's where you could nonetheless measure them. You measure them with your ruler, right? And then once you're done, you actually take out the seed, because the next step is actually to weigh them on the mass balances, where you're going to be taking their units into gram. Now, some of you just want to let me know your conditions. Normally, the weight balance is already there, so it's very simple. You just after dissecting or collecting the shoots, right, or the seeds with their shoots, you snip out with scissors the seeds, or you essentially just literally take it out without the seed, but have the shoot placed on top of your weight balance once you've tuned it out. Some of you may have to zero in your mass balance, so just try to do that in advance with your weights. Good. If you're finding having difficulty operating, also let me know. Some of you may have real that's good. So just turn them on in the meantime for your mass balances. I know some of you have the mass balances, others can already begin measuring. So in the meantime, in the next, I would say half an hour, you should try to do all of them, both the observations, the quantitative observations of biomass and of shoot length. Any questions? Any questions? No question? On your mark, get set, go. So you'll be essentially making note of these observations for your shoot length and biomass on pages 43 and 44. So what I recommend everyone just to pay attention is that you're going to essentially, group 2A, want to make sure that you're paying attention. Pay attention. That, and likewise control treatment A. Control treatment A. Please make sure you're paying attention. So that way we maximize our efficiency and hopefully you can come go home or leave the lab earlier. The goal is that to make it efficient, you have 4 people in your group. Try to split up tasks if you can. If you find it as a group that you're able to do things quite easily, like a factory machine, then what I recommend is just doing this as efficiently as possible by accounting for shoot mass for one seedling, and then the length, the length first, and then the biomass afterwards. Just try to do this as simply and as accurately and as efficiently as possible within the next half hour. So by 11:15, for 20 observations of both recordings, it shouldn't be too difficult. But you can begin, just bear in mind that you are keeping track of time or someone in your group is. And if you have questions, just let me know. Thank you.
So, follow 20 things. Make sure you get like almost yeah. That's fine. Just follow 20, but it's just that. Well, try to hopefully. I'm fine with it.
We're pulling it out cloud arrays.
Some of you may have seedlings. Some of you may have seedlings that are attached to 2 shoots. If that's the case, you have 2 options. You can split it in half, and that way you have 2 separate counts. Or if you want, and you have a lot of shoots, you can actually just find 20 different seedlings and then just account for the longest shoot for your measurements, both for length and biomass. So let's say you have, some of you may have, let's say, essentially you may have, let's say simple. Yeah. Then you have to either include it or if you include it, you have to definitely include it for but if an optimal that is too small, you might be able to be read on the mass balance. So if that makes sense, I would simply just count consistently for yeah.
When going through watching, do we have to identify which side to pick it from?
No. You don't have to identify the sides that you're taking the cross sections for the seedlings. Just bear in mind that you're taking 7 randomly from one side, 6 from the middle, and 7 from the other side.
Yeah, just do it in centimeters, maybe convert it. Like you can see. There's probably 2 people measuring this. So what do you measure? Give me bumpers. There's gonna 6 people.
Any questions? Anyone?
Okay, yeah.
You're reading me out. So we're just going to shoot not zero the.
Yeah, you can
But
do you have a third ruler? Can I take it? So I'll do the last subject. Yeah. So I'll do 20 and 13 will be fine. Right, because I got the right.
Yeah.
Yeah, I'm just counting the line.
yeah, but make sure you guys don't. Yeah. Yeah.
Just a reminder, everyone, if you have more than one shoot, make sure that you're taking it out for your biomass. You can take the longest for length, but make sure that you take out the the longest one, or isolate it to seasoning accordingly.
For this one, I would take all this part on the back because this one's at 1%. And then I would go
Into it.
I'll do this one.
Any questions? Yes. So if you get 0 on the reading, you can place it at 0.01.
Oh, yeah, that's what we're
Oh, they're waiting on this one. They won't have it order.
This one, and when I removed it, what it's showing up.
Just leave it.
So it seems like you got a bit more.
Yeah.
And that's interesting. No, I think that's like the woods.
Yeah.
So if, by the way, bear in mind your observations even quantitatively. If you find that you're seeing, for example, the mass balance moving in numbers like simultaneously or abnormally, or you get negative balance and you're concerned, let me know. Also, you should let me know how you're actually, or ideally make it consistent in your how you're measuring the different ceilings and your length and biomass. So that way you're consistent throughout the remainder of the mark. And also for measuring your control variable.
But we don't want to see. Yeah, I feel like they're all going to be about the same. Oh, yeah, you guys want to come here? Oh, you can see the map. I just take the photo. What you do? This is great. No. Not that sure. Oh, not that. Oh, yeah. I don't know. Double. That would probably be the longest across the board. Yeah. I wouldn't say we don't, that's a pretty great. We have not made a small one. Yeah, that's right. Yeah. Okay, so it's like. We're just saying. Because we'll see that happens. Yeah, most of the these are the trap. These are the these are all that. Which makes sense because it's just very noisy. Not a bit. That's true. Yeah.
All right.
Except for that three.
Oh.
I don't like all of the ones that have. Essentially. Keep it up, you guys. Don't worry in terms of like comparing yourselves. I just have a question though. So control treatment B, the second group, how many recordings do you have in total for your length and biomass? 19, okay. So you're almost done for without the biomass. Okay, so just a reminder, make sure for biomass you take out the seed, and if you have two shoots, take out the lower one, the smaller one. Treatment 1B, how many recordings do you have? In total? Okay, that's okay. That's good. If you guys want as well, you can maybe separate as groups, having some of you focus on the length and then quickly giving it for biomass.
Yeah, she's doing the light now.
Okay, okay. That sounds. Then maybe if you haven't taken the report out, then maybe actually. Just do how many recordings do you have? Okay. How many are you bear in mind that when you're done, just let me know as your group completes their component. Also, if you haven't observed other members' qualitative data for the other treatments, this is your time to do so. If you have a spare member that can make a note of other groups' observations of their, essentially the observations we talked about earlier, the colored dots, the patterns that you find in your containers, that would be very useful for your FLR. So, or at least for your manual. So just bear that in mind. Try to complete that section as well. And it could be just one member of your group that focuses on it to grab and collect that data. Perfect. I'll make note of it if you don't mind.
Yeah.
Let's see. In the meantime, members of your group can start cleaning up the area. I'm just gonna input them with. Okay. And so,
Put them in back to that.
just a kind reminder, everyone, you have under 20 minutes. So I'm giving you extra time, or at least I want you to be mindful of time, because we're gonna have to go to the last exercise for today. So try to budget your time wisely. Work as pairs if you're feeling a bit behind. If you haven't reached halfway as of yet, I would recommend separating as groups, having people that focus on biomass as pairs or as singlets, and then others that focus on the length. Thank you.
Can we start experiments too while we wait?
You actually can. In the meantime, though, so I would prefer to give instructions to everyone, given it's a bit, let's just say a bit technically heavy. But that being said, definitely you could review the components because you'll be able to excel proficiently. Now you are treatment 2A, right? So let's go to the top. You can give me your data for the length. Perfect. 85. 115. Okay. 75. If you want to even read them out to me, I could copy them.
Okay. Let me do that now.
If you can.
Where are you at?
76.
87, 71,
71,
100, 50, 115, 103, 104,
115, 104, or
103, 104,
103, 104,
87,
87,
85,
85,
72.
72. Okay. And
You want in grams, right?
yes.
Okay. 0.05, 0.04, 0.07, 0.05.
I mean, I'll let you know when I'm at.
Bishop, is the one after 105, 160?
Was it? Maybe I read it wrong. Okay, so 160 is 100 for the line.
Okay. Okay.
0.05, 0.04,
Oh. Okay.
0.04, 0.04, 0.06, 0.03, 0.04. Where am I? So
We're at
0.03.
0.03.
Yeah. 0.04. 0.01, 0.03, 0.05, 0.02, 0.04, 0.03, 0.02, 0.02.
Congrats. Great job. I just want to share, if your group is done and like you're waiting in line, you can actually continue looking at exercise to prepare yourselves. Yes. Generally speaking, yes. If you have them all, 20 of them in one location, you could just count them all together. Gloves, you could put it in the garbage. Thank you. Let's see. Before we continue, I just want to make sure time. How many of you are almost done? At least completed one biomass or length assay. One of them, at least one of them.
Yeah.
Okay, so you guys are almost done. Are you guys almost, how many more is left? Your balance? Okay, give me one minute.
Yeah. Yes. I can be for every. This is really. Design tissue.
Yeah. Which one do I say first?
You can do that.
Okay. 76,
6,
84,
84,
123,
23,
85,
85,
77,
87,
82,
82,
87,
87,
132,
132,
126,
126.
141,
Are you sure 141? Yeah, like 1 to 14 minutes. 41,
104,
104,
89,
89,
93,
93,
106,
106.
179.
That one I would actually ask if you could read them.
Really?
Yeah.
Okay.
The reason being is because if
It's just too like
it may be an outlier, that's
Yeah.
why.
Okay.
Like unless you've seen multiple that actually look that tall,
yeah.
like you haven't read the entirety of it,
Yeah.
but do you think there are a lot of them that like look like that or no?
Oh really?
Yeah. Can you just read that one? That's okay.
Yeah. So take out like do now or like
If you can.
okay.
Or actually I could take the rest of yours.
Okay. So no 100.
Maybe ask someone to do it in the meantime.
Could you measure another graph?
Just one more.
Just
Just
one
one.
more. Okay. Yeah. So continue.
Yes.
132,
2,
139,
139,
104, 143, 151. Yeah.
Okay.
And then the gram, right?
Yes.
0.02,
Just before I do, I just want to make sure I have everything. 0.02. Yeah.
0.01,
1.
0.01, 0.01, 0.02,
2.
0.02.
If you find data that also replicates multiple times, let me know and I'll just copy and paste it.
Okay. Well, the next one is that.
0.02.
Yeah.
Yeah.
And then the next one is 0.02.
Okay.
And then 0.03.
Yes, yes, yes. Yep.
0.04,
4.
0.01,
2.01.
0.05.
This is also number 12, right?
Yes, that's number 12. 0.01,
2.01.
0.03,
2.
0.02,
2.
0.01.
0.01.
Oops, sorry, 3.
That's okay. 3.
And then 0.01,
0.01.
0.01,
0.01.
0.05,
0.05.
0.02.
Okay.
And then for number, that was 15,
right?
yeah. What was the length?
79.
Yeah, 70.9. So
7.
wait,
So,
Oh no, like wait, can we get like a different? Yeah, because it's like too tall.
if you want, actually, I don't mind if you write it in two groups. It's okay.
Oh really? Okay, okay.
To make it easier, and I'll just also keep an eye. Joshua, sorry, did you have a question or were you done? Perfect. Okay. I'll take his results. You can also help your team.
Okay.
And then when you're done, when you get the result, let me.
Okay.
No problem. Do you want to even upload them? I'll just check on other students, but for context, we were the middle row of B. So exactly. And that would be for, this is essentially going from shoot length, and that's for biomass. Thank you so much.
No worries. Thank you.
I also want to sit down.
It's all good. I won't take your seat. It won't be too long. Doesn't copy the decimal place. Thought I could cheat.
No, that's okay. I'll help you. 0.01.
0.08.
0.08. For all of them?
No, just for this. It changes for all of them. I wanted to see if I could copy the decimal of 0.0 and then just go 1, 2, 3, 4, 5, 6 sort of thing for the numbers. But it doesn't copy the zero, so you have to type it
You know what I do? I just point that you put the dot, decimal, and then it'll go. But here's what we'll do just before you go forward. I know she had 115 and 0.04.
Yeah.
115 and 0.04. So I actually did dot for period 0.4, and then I get you decimal. I mean, you're pretty much putting 3 values for each round or each set. So if you want, you can continue.
Yeah. No, I just want to see if I could copy like the 0.0 and then I could just type in the number. So for this one, how I could just type in 2, 3. I gotta go 0 and
Here's what we can do. Put them all on this line, and then.
well, I mean, that could be good. I can copy.
Sorry?
I can just put in these values, can't I?
Yeah, you can.
Okay.
Both ways work good. Yeah, you can go the easiest way.
Sounds good. Thank you.
Yeah, so I just work at Walmart and you get 20% off. Classes are already like $400 off. So I got like so I'm not listening. As you so let me show all of you guys the kind what we need to do for exercise 2. I'm going to show it to you as well eventually. Actually, if you guys want, if you're anyway to complete it, I will so
Yep.
what you'll be continuing to do out here. Now, the idea is, has anyone seen or used the microphone before? This is- So really, this is where like integration comes in again. I'll tell you why in brief timing, but pretty much what you're doing today is making use of all of the poetry or so you have like styrofoam boards that you're going to cut off and essentially fit in the celery stalks inside your microphone. Microphones are used to essentially cut very thin slices of specimens of material to be able to fill at them under a microscope, which I believe each set of yours should have. So you'll be able to use that for today's practical to visualize different cell types within the celery stalk. So that's your celery stalk. Each bench is going to use one set. And we're going to try to find tune one as an example, so that way you can all be able to get some really nice cross section. Each group has to do 5 for your analysis to be able to take measurements as well from cell size. So just bear that in mind. You have 5 cell types in total, and one of them from table 2.4 you won't actually need to do. So just bear that in mind. Your materials are the following. Anyone can't see, just let me know. The following, and you'll find instructions here. So if you have any questions, if you want to revisit them in the lab manuals, but I just want to bear in mind the following. If you don't have petri dishes, yeah, perfect. You have petri dishes. So you should have petri dishes for water, and perhaps as well TVO. TVO is under veggies. It's small, dark brown looking vial. And so the idea is that's where you want to have gloves on, as well if you have like allergies to plants, the grass essentially that we need to recreate. But essentially, like you see in figure 2.4, or one of the last figures for data observations, you're going to be making thin slices of what looks like this first little. Now, before doing so, you actually take this out, and it's shown also at the very bottom of the picture right here. So take this out. This is where you can use it with your gloves. But the idea is that you're unscrewing at the top to be able to allow or to increase the amount of space you have to fit in the celery stalk. And this moves backwards so that way you can give it more space. Think of it like the cabinets that you need a given amount of space to successfully put in your luggage or your belongings in, and this is your luggage or belongings for too. So now that being said, obviously you can't like fit it in, right? It also has a sturdy like alignment here, so it's very difficult to fit it, right? What you're going to do is the following. You're going to use your razor blade. That's why you also want to be holding or having gloves with you as well. There is the tilted or tilted edge where you're holding, and not by the sharp edge where you're cutting. So just bear in mind that you're going to try to make. It's going to be all able to see this. You want to come in closer, you can as well. So the idea is that you're going to make a cut such that you know it should be able to fit, right? Because for me, I know that a given amount should be able to fit inside, but everything else may not be able, right? So you want to estimate. You may have to cut more, but the idea is that you want to take out any loose edges that may essentially constrain your cross section, right? So that's okay. That's good how it is. Now it is quite long as well, and for context, even though I made it, you just know some of the selling salads. You're gonna have some bits for lunchtime. Now, this is how far it goes. But the issue is that when you begin at least, when you place this back inside, obviously you can't even fit in here. The idea is that you have a thin amount, and normally I don't want you to have to do this, but generally speaking, for a first round, you have to cut, and you can actually take it out and repeat the process so that we straight. There is the other caveat I just forgot to mention that you want to actually cut side that's already painted. Who knows why? If there is a straight side with the stalk, and there's others that are more that cut obscurely. Why would you want the straight side to be inside and the one that's painted to stick out? Like at an angle. Maybe. Not necessarily, but the benefit of having it straight is that you're able to not only fit it in correctly, but so that way it's straight at the very bottom, at the end of the microphone. If you don't have it as such and you have it inverse, then as you keep cutting and as it gets more fragile, you run the risk of it actually not making straight across. You just want to actually, more importantly, maximize the stability of the microphone cut because it's going to go down like a essentially almost slant, slope, and then eventually cut through. So I'm going to repeat that process, pretty much cutting this, once along its length, and another time, assuming it's out, right there. That should be a good cut and good enough for you to be able to take very thin cross section. Any questions? No? And just to confirm, everyone has placed in their values? No? Okay. You can continue if you have time. But before I end off, pretty much now you want to get your luggage fitted in nicely, right? The celery stalk is what I'm referring to. And now you will essentially continue to keep it tight and frame, but not too tight such that it's difficult to take out or comfortable. And then you slide it in. You slide it into this this microphone, and as you can see from the manual itself, or the illustration, this sharp edge is where it's actually going to get cut. So I'm going to just show this. If anyone wants to go on that side to see it actually get cut, but let's have an idea of how this. So that's your thick edge. That's a thick section, and that's good how it is now, because obviously you want to play with your first section to make sure that you're getting it in accurately, and then once that happens, if it's actually still embedded, then you use the paintbrush. Don't use your active hand to take it out. And then you pretty much can repeat the process of either pushing it in further or taking it out, then spoon, and repeating the process so that way you can actually take that 5 fine sections. I'm just going to do this one more time. I know this may sound a bit more longer or may appear longer for your practical, but this is one of the final parts for today. And then you have your maths. Let's finish this off. So let's say I made this one maybe a bit smaller because there's a amount that this one could go to. You can also just artificially move it forward, right? Maybe just circle it. And then clamp it down. Place it all the way down. And then you repeat the process again. So now that it's on the side, you should ideally get thin, very thin slices. Just be mindful. I know I'm like focusing a lot on the microphone, but yeah, the razor blade, you want to make sure that you here or any questions going on? I know it's a bit hard to adjust that, but if you have questions, let me know. Sometimes within these experiments, essentially, you may even want to just do it manually, such that the thin slices come out accurate. So if you're having difficulty, you can also use the tweezers, right, to recut and readjust. I'm just going to do one more. Yeah. If it's a phone call for your parents, you can ask. Section 8. So this is exactly what we want actually, and it can even be smaller. That's okay. But this is what you want to get. Essentially, you use the creases to pick it up. Just bear in mind that it's a bit fragile. So when you are placing it into your petri dishes, you should have one that has water already filled in. If you don't have water, it should be placed into the petri dish from one of the distilled water tubelets. But other than that, you should have the following second petri dish, so the outer side, sorry, the other side, with TBO. TBO, you're only going to add, even if you don't have it on the surface of your petri dish, you'll just add it on top of your celery stalk cross section, and just make sure you keep track of time. 30 seconds is good enough, and then you should be able to rinse it off either with water, each section, or each set of sections, well, and then, or just what I like to do is just place it into that other petri dish with water and just stir it off, so that way you're able to take out and rinse away the water, TBO, the residual TBO. When you're done with that, you should have a covered slip. Just bear in mind that your celery cross sections, all 5 of them will look purple, okay, on the reactions, and then you place them onto the slides. You can add 2 or more depending on the cross sections onto a given slide, and then you can be able to take observations and measurements of them. And that will pretty much nearly finish off the lab. Any questions? And a kind reminder, please to use the phone, especially when we're doing instructions, because you want to maintain focus, ability, and increase or maximize efficiency throughout the lab. Lastly, remember you have 5 sections. Because you're dealing with TBO, bear in mind you're also wearing gloves, and that you try to do this as efficiently as possible, meaning that you have a petri dish that already has water. You have a petri dish that can even be left blank without any water, such that you're adding in TBO, and then when that 30 seconds are done, with TBO staying on top and across your surface, just on the celery stalk itself, you're able to lift it up and take it out for essentially rinsing off with water. You alternatively can also just rinse it off on the same dish, but I would maximize efficiency by using the other side of the petri dish to rinse it off accordingly. Any questions? For timing, question? Any questions? Okay, so for timing now, in the meantime, I would say everyone who hasn't uploaded their values, just have one member of you do this essentially, and essentially upload your values. If you haven't done it as of yet, just wait in line, and then in the meantime, other members of you could focus on taking the cross section. Yeah. Yeah, question for this, if you as I did before. English water. Like all 4 or 5.
Is it group of 2 or group of 4?
Group of 4. Each group will have 1 celery stalk. We have 6 of those, and make sure that you're working as groups together. Just bear in mind that one member of your group at the end of today's lab should have added in your values for the true biomass and of the length.
So I guess we can cut out the
So
One of the last video. Thank you.
That's it.
This should be the one.
This is good.
Put it into the water. Yes. So that way you can click this. You can see them. Yeah. Doing anything. You can take. Anyone having problems actually fitting in the celery stalk into the microtome? No. What time do they pull it up? Oh man, that's so wonderful. Yeah, I'm doing my camera, so we do this right. Yeah. It takes all the way. Yeah. Here, you want to do it? Make sure once again you don't put your fingers through the blade, either the razor blade or the blade. I feel like you just go to the board. You always just do that. Wait, let me see if I can get it. But there. Yeah. You can have thin layers of cross sections because otherwise it'll make it difficult for you to visualize. When you're done, let me know if your cross section. I just want to show everyone this is a really good section. The one that's helped me take it out. So if you have yours, that's good. That's good as well. Just have them as thin as you can, even if you need to cross section with the razor blade on the signal that you have, that's okay. But try to be done within the next 5-ish minutes. Ready?
Oh, that was really fun.
That was a lot easier. Yeah.
Keep it up, you guys. Oh, please sit back when you're done.
I would.
When your group is done, let me know. And you can also dispose of the extra celery stock into the garbage or into the containers that you have
Okay,
your
yeah, so
plants.
we do one at a time. We just put all five in, of course.
So if you can put all 5 together, I would, like, of course, some people take them all out.
Oh, yeah.
So the extra time that it takes, is what time that it takes.
Yeah. So we'll do like an assembly, like you take it from here, or
Also, to put in all 5 is not, essentially, you
yeah.
If it wasn't,
Okay.
but, the idea is that you would then be able to make, 5 inside, you know, so it's okay, we could keep them based on the quantities you have, and each member would be have a.
Yeah. And right here. No, I'm gonna put it in here. You're gonna pour a little bit.
So make sure, once again, as groups, you're devising a unique plan as to how you're gonna take out TBO, but also take out,
We're gonna give it like 30 seconds.
TBO, containing celery cross sections and place it into water. There are different ways.
I think so.
If you're having problems or difficulty doing this, let me know.
Okay. Let's do that. Yeah. I guess I'm gonna wait. You want us to just pause? I'll go over there. Could I go wherever you are? Oh, yeah. Yeah.
Essentially, once you have all the then
Okay. Yeah.
Oh.
Yeah.
So by about 12 o'clock, I can help you guys obviously prepare, but just let me know if the group has easier help making the process.
Whenever you want to do some of that. No, I can see it pretty well. Looks like a. No, it works. Where are you measuring? Really?
Understood. So, just to clarify for everyone, once you're done with 5 cross sections, let me know, so that way as a group I can give you more instructions, more.
Okay, so you need one measurement of each. Okay, we check this. Okay.
So, in about 2 more minutes, I want you to complete the rest of those processes.
Okay, sorry. No, it's the average. Yeah, that. Five. It's just that. Wow. I'm just gonna keep it on the slide.
So,
I take it.
I just want to say, everyone, you guys are doing pretty good for timing. Great job. I just want to also share now that many of you are almost done, all 5 cross sections. Make sure that you organize your location. Make sure that the celery stalks, you place them back into those containers because you're going to take them all together to discard or to like remove and then return them into that clear tray afterwards. Now, I want to make sure a few other things. So let's have a look first at your results. Generally speaking, shoot lengths shouldn't be over 2 meters, 2 centimeters. Oh, sorry, 20 centimeters.
200 millimeters is what I'm referring to. So I see one here. I think I saw the students who said this. At least for the control, it's reasonable, but let's see others. That should be okay. Group 2B. You don't need cover slips? Before I see 0. Is this 0.1 or 0? You are 2B. Okay, sorry. Thank you so much. Control 1B or control B. I see you have value 0.1. Is it 0.1 the size of your tube biomass, or is it 0.01?
Hours. I did a mistake where I put hour one in B instead of sorry, A instead of B. So I
Okay.
think this one is
There?
yeah
For both or just one?
for both.
Okay.
Yeah.
Okay, I'll ask them then. Okay. Quick question, you guys. Do you have values that are 0.1 for your biomass or 0.01? So it actually went to 0.10 grams. You saw that from the mass scale? It was tiered as well? Yeah? Okay. We'll keep this then. Now, question.
You mentioned that we should take it out of the water. I don't think drop it on. We only have to two. So if I put it back on the original tray in the back, this
In your
Yeah, we've actually, because like I feel like I'm touching here.
Still right.
Yeah.
Maybe let that this.
I think it's good. That works. Look at this.
Yes.
So, how's everyone doing now in terms of timing? Yes? I can help you guys shortly or after a few announcements. In terms of group 2 on this side, are you guys now doing visualization or have you already stained your celery stalks? Okay, so you've stained them all? Only one of them? Okay. Have you stained all of them? That's okay. That's okay, by the way. Don't worry. Have you stained them all? Group 1, control A. Control A. Have you guys stained all of your celery stalks or cross sections? Sorry? In the process, okay. All of you, I believe, have you visualized them? Yes or no? Are you staining or visualizing? Are you staining with TBO now or looking under the microscope? Okay. So let me just share some important points. Everyone, I just want to make sure you're paying attention. Here are some tips for those that are already in processing your cross sections. If you've already done this, that's okay. But it's important that everyone else present to be aware. Now, you have, many of you, 3 different dishes, right? One is plain, or actually one is, I think, like yours was, yeah, with water.
So you have one that pretty much is suspended with your cross sections, right? All 5 of them. Right? So let's just put 3, for example. And then another that's usually just left empty, and another that has H2O or water, right? Like it's recommended that essentially you have a body of water, so that way when you either in this or in the second plate, you are able to essentially add in drops of TBO. Some of the markers are a bit off, so don't worry. The idea is that for those that are in this step, you can add in at max 2 or 3 drops just to fill up the surface of your celery stalk for each cross section. When you're done 30 seconds, that's where you, up to a minute, but I would say 30 seconds better, to then after transfer that stained cross section into that body of water and then rinse or stir your plate with that well, so that way you're able to remove any residual water and then repeat that process for the remaining ones. Now, some of you have asked as well for your cover slips. What do you do? So, cover slides, like your slides, testing for visualization. You can add probably 2 to 3 of them, depending on their sizes, right? But let's just say these are 2 or even 3 different pieces. You can add them all because it would have been added already to water. You don't have to add secondary water droplets on top of the specimens or cross sections. You can just view them with their stain as is. Now, when you are at the step, you're going to be visualizing. You're going to take note of the different cells, like making a drawing of them for the different conditions, right? We have 6 different cell types, generally speaking, in angiosperms and in general, like in plants, like the type that we have here. But it's important that you're able to note which of them exist in the celery stalk. So bear that in mind. There is one missing, but you have to bear in mind that we complete the following 3 columns. And that is, you draw what the cells look like, right, under the microscope, right? You'll see different, essentially, cell structures with different sizes, right? Be it the parenchyma, the parenchyma, the xylem, the phloem, etc. Right? So just bear the different structures, the different sizes, the different locations, how they appear, how adjacent they are to the cell wall. And bear in mind, are they primary or secondary? What's the difference between a primary and secondary cell wall? Does anyone know? Or could anyone make mention of examples? Let's just go back. So, for example, and actually these are following slides, so don't worry in terms of these details. We already completed the FLR, right? The data collection of the FLR. And then afterwards, it'll be uploaded eventually on Quirkus. If you have questions, this is where you can ask them to the different BioHelp emails. And as well for sessions upcoming to prepare yourself for the writing assignment, right? Which was moments ago. But also you have for the writing assignment part 2 for instructions and further details with office hours for both Mark and Andrea. So you can bear that in mind. Now, exercise 2 is where I was talking about plant vegetative morphology at the beginning slide of today's practical. So this is where essentially that diagram of what I was drawing comes in to your microscopic analysis. Plants, as we know, they have different functions and have different requirements, right? They need light, oxygen, CO2, nutrients, and also water, and some of them just use soil. There's also the mode of hydroponic plants, so that's an extension for those that are curious. All of these different components and elements are involved in photosynthesis. Many of us hopefully know the chemical formula or just recall learning it in the past. But essentially that helps to develop energy and carbohydrates as well.
vegetative organs, tissues, and cells also allow or help plants to receive, transport, and retain these different elements. So bear that in mind because that's pretty much what we're focusing on today. And that's where, under the microscope at 40x, where you're going to be looking at now the different tissue types. So now within the celery stalk, there are 6, technically 5, different tissue types or types of cells that you'll find, right? I mentioned them earlier that you have as examples based on different morphologies, the range, size, the shape as well. You have parenchyma, parenchyma, you have xylem, phloem, right? So bear that in mind because that helps you not only prepare for completing, as we've done, the microtome and staining, but also to be able to hopefully measure other readings, right? Like your cell size. Now, I could go through this in due time. And forgive me if I just went straight ahead. For timing, I also want you to have enough time to complete today's lab. And if you have questions, I can do my best to help. But remember what we did from lab 1, right? Calibrating or actually measuring cell size from the pollen grain diameters, right? If you recall that, the idea is similar because as you can see with this example of a pollen grain, that you're able to measure, and this should help, I believe, John and his group answer the following question. So let's say you have a pollen grain, right? 15 OMU, right? Because you're going from 10, which is okay from your eyepiece, going from 10 to 25. You don't have to always start at 0. But you have the 15 OMUs or ocular micro units. I actually go by OUs, ocular units, to make it easier, but the point is very similar. Under the microscope, you have this chart. You're at high 40x mag. So you're going to multiply pretty much what you see, the number of OMUs, which is 15, by the calibration factor. When you multiply OMU by micrograms, microns essentially divided by OMU, the total unit is going to give you micrometers or microns. So essentially you're doing 15 times 6, that's 19 micrometers. So bear that in mind, you're multiplying the two, your units of ocular units you find of how long or how large the cells are, and you multiply that value by the calibration factor, which is at the very end, the last column, and you have to intersect with the magnification you're at because different magnifications have different calibration factors. Right? Because the OU, the eyepiece itself, the ocular micrometer is going to remain stagnant in size as you go from different magnifications generally. So what you want to bear in mind is that how is that calibration factor then changing? Because if one setup remains constant and you're increasing the size of as you go into greater magnification, you need to adjust accordingly the calibration factor, which is why I'm saying just to repeat it one more time. If your pollen grain, or in your case, a cell size is 15 OMU, you multiply it by at 40x 6. 15 times 6 is 90. That's your size, 19 micrometers. Make sure you use units. Microns or micrometers are okay. Any questions regarding that? No? Please make sure that you're paying attention as well, because I could tell if you're not. And I always want to make sure you can all succeed in this class, right? So just do your best, right? And leave the rest with your best. So that's the best advice I could give just before you end today's class. And then there's lab 2 as our assignment. Before we end off today's lab with the final assignment, with the post lab as well, bear in mind that you clean up the arrays, the assays that you use today. So for example, you're not only shutting off your microscope and turning it off, right? But you're also setting off the mass balance, you're disposing or returning essentially the plant biomaterial into the trays to be essentially, hopefully reused in future practicals. The containers can be placed in the Go Here icon or that paper beside essentially on the left-hand side of the sink So just bear in mind that you return the containers that you used, untape, remove the tape, and place them into that clear bin there. As well, make sure that you take your personal belongings, make sure you wash your hands, and have a great day before you leave, right? Now, lab, we're almost done the lab, really, right? And so I just want you for the time being, next half an hour to, or even let's say 40 minutes at max, to start taking those values. I want to ask, let's see, you have this, if there's anything else. Yeah, this is relatively simple actually. So try to do it even in 30 minutes, right? I'll let you know in terms of timing. By let's say 12:37, you guys could be done. But if you need if you have questions, let me know. The color is what you should see quite clearly. Some are purple, some are bluish green. So bear that in mind. Some are purple slash pink. That indicates, as you can see in your lab manual, one type of cell wall, and then there's the other that is essentially the other form. Bluish green is for one set, pinkish purple is another. So just bear that in mind. Look at your lab manual because I want you to be able to focus, being able to understand the concepts we've talked about in the PowerPoint, but also focusing on what you've done and hopefully answered from the manual so that way you can excel in future quizzes and hopefully have excelled in the previous one today. Any questions? Any questions? Yes. Yes. I see. Yeah. Yes. Yes.
What if you, like, you swap to like a slide, something so we could see, or whatever, it doesn't matter.
It shouldn't be a problem. Believe it or not, let's see if I switch slides. You won't see it like this. This is as almost as close as it gets. I'm gonna show an example soon, but the issue is it's part of the answer key, so I have to hide the slides. So, let's see if I can draw. I'll draw it to make it easier for you guys. No problem. So, just to answer this question for many of you, if you're trying to differentiate, okay, what color is the stain, or what color is my cell wall, or what type of cell wall am I looking at, and how am I going to differentiate the cell types that I have here, only amongst the 6 that you see. The lignified portion, right? If you're looking at the very top layer, or at the very top of your cross section, your celery stalk, that is essentially where the lignified portion comes in, right? And so that's already giving you a hint, right? Remember, cellulose plus lignin, which makes with the TBO stain, appear your cell type, makes the cell type appear bluish green. Let's say even green here. Greenish blue. Turquoise. Essentially, that's going to hint for you your secondary cell wall. So bear that in mind. Everything else that is pretty much purple, pink, is pretty much an indication of your bluish, of your primary cell wall. So, like, I know these are, okay, I drew it in a different way, but the point is, you have some cells that appear in clusters like this, much larger, right? It's shown here beautifully on the slide. Primary cell walls, say pinkish purple, right? And then there's others that even in their smaller sizes also appear pinkish purple, with the TBO stain, right? Following that rinse. So what that indicates for you is that that's your primary cell wall. Questions for you, sir? No questions? Okay, don't worry. If there's questions, let me know. But that primary cell wall is made up of cellulose as well, but plus pectin. So to make it easier for you, I don't have a purple marker, but the point is that purple or pink, purplish, purple, pinkish hues. Like if you see pink and purple, I'll start with P. And essentially, pectin also begins with P.
that indicates your primary. I'm using like biochem notations, but that's your primary cell wall. So bear that in mind. 2 with a dot top indicates your secondary cell wall, right? So just bear that in mind. Cellulose plus lignin in your structures of, let's say, different cell types we touched upon thus far, and those you see in the lab manual, that indicates that there are secondary packed cell walls, whereas those that are with cellulose plus pectin and depict a pinkish/purple hue following the tibial stain, that's your primary cell wall. So make sure, especially we're going to have this in your lab 2 assignment, do 24 hours from the end of today's lab. So bear in mind that you make those points readily understood, or at least highlight the different traits I've touched upon thus far. As well, you have, you're going to draw, right, 3 to 4 typical cells that you see for each cell type, but you don't have all 6, you have 5. So bear in mind, based on what I've touched upon, right, there is a bit of a hint as to which of the following, if you look at your lab manual, and based on the different cell wall types that exist, primary and secondary, and which of the following fit in either or, find out which of the ones are missing. I told you that, like, think about critically what I talked about actually. That way, you're being it. Any questions now thus far? Any questions? So I'm going to give you until 35 to finish it. It shouldn't take too long because many of you should have already been able to complete the first 2 categories or 2 columns, or at least are halfway there. So now really in the meantime, if you need help, think about what I talked about earlier. They're also shown here. If you have, let's say, a cell, even if you have multiple cells, I'll even draw it there with the following example. Let's say this is your OU, and each division is essentially read like a ruler. It goes up to 10, and then you essentially divide accordingly. So let's say this is 0, and this is 1. If this is 1, and I have 2 cells here, that means it's 0.5, right? OMU. If I'm at 40x, I'm going to multiply by 6. Okay? So just bear that in mind. At 40x objective, a short form, you're multiplying by 6. 0.5 times 6 is 3, right? So that's 3 microns or micrometers. Bear in mind that you use those units, right? It's 3 micrometers. And then you're essentially repeating that for all of the cell types. Now, I want you to bear in mind that in the next 6 minutes, roughly, I'm going to ask each group how many cells you've recorded. So bear in mind that you're keeping up with pace. I want to make sure that you're able to complete the lab easily, but also that way you have more time to even complete your lab to assignment. So bear that in mind. If you have questions, let me know, and that way you can also help others before they have to before the next lab. Like, what are any questions? Yeah. So when you look at this, you'll see like a big cluster of purple in the middle. That's so I recommend using all of your cross sections at least 2 to 3 on one slide. Don't use all 3 slides. You can technically use 3, but use maybe 2 slides because you can fit it 3 per slide. So that way you're able to just move left and right, take as many readings as you can, and so in case you find one cross section missing different volume.
I assume it's this one that's not present. The set. You said one of them we want.
This, they come like a green.
Yeah, like, kind of, like, I don't know. You know how to do that. I think so. It's present, right? The other one, like,
That's what I was like,
And then, so it's very simple. Once you're looking at, like, let's say 5 different cells, you should already know then after what the cells look like. Finish the first
Oh, that's pretty
column,
good.
for this table on page 37. And then the color can also be noted down. So just bear in mind that you're taking notes of the different cells now, and then hopefully in the next 4-ish minutes, I should be able to put notes how you should be able to do this.
Yeah. Okay, I just go through all of them.
Remember, there's only 5.
So what does it ask the?
Which one are you?
You got that? Sporting doesn't work, you can't see.
Okay,
Yeah, I don't, I think. Like, that's a Google. You want to put it in your slide? That's a pretty good.
So continue as you are. I'm going to start asking go in terms of your measurement of how many.
I think they all look so good. Yeah, 40x is hard to see. I when we did calculate, like, there's no like you can't really see the difference between themselves. They're like, yeah.
Yeah. I think it's good. It's good as it is. Just I would say, before I go to the lab assignments, actually, I agree with it's 45. It's 45. I'm looking that's pretty much 1 major component of so now let me ask how many recordings I should ask them. How many recordings?
So aside from the three.
Yeah.
That's really good. I did not. Some of this is good. No. Yeah. Okay, okay, I got it. Five hours. I know. No. I'm good. Yeah. Right. Yeah. No. Yeah, I think it's not good. I think it's literally that's PowerPoint. Yeah. There's nothing I can see.
Now, I just want to ask, how long would it take so just so you guys know, this is the lab assignment. As well, I'm going to show it again in about 5 minutes, but if you wanted to have a look, you can. Are you guys just cleaning up right now?
Perfect. So, let's see. I think you guys are good. If you just shut off the microscope when you're done, the mask balance should clean up the visibility, making sure everything's clean. Yeah. TBO. Very good. Yes. So, great question for your glass slides. And by the way, before you're done, I recommend taking photos of your essentially your cell types because you're going to use it for this lab presentation. For the lab assignment, you have a presentation to look at 3 cell types and to focus on the different conditions mentioned on page 45 and also shown here. You want to provide details in 3 minutes for all 3 cell types, just a minute each, talking about the arrangement in a PowerPoint, the thickness, the cell shape, the abundance of tissues, and what not only the factors, but what functions those cell types have. So some are involved in the movement of water. Some maintain the stability of the plant itself. There are different functions that they have, and you could see them from your lab manual. So just bear that in mind. But I want to make sure that you're able to complete this section, the one that we were just on, right now, essentially. So make sure within the next 8-ish minutes, because by now I want to know that you guys are already complete the first 2 columns, right? So remember, there's only 5. Some of you have already noticed this, that there's a parenchyma, right? There's essentially 4 on one side and 1 on the other. So think about that for a moment. How many cell types do you see pink or purple? Are they more than 1? Is it just 1? If it's more than 1, then you think probably that it will probably be the other option, that's bluish green, that will only have 1 structure. So bear that in mind. That may hint to you which of the cell types are not present. So bear in mind that you're able to make note of the different ones. Hopefully you've drawn already by now or in the next 2 minutes. Please draw all of the cell types that you see. It could be just 3, it's okay. Some of them have different cell arrangements like the ones I showed there. That already is like about 3 or 4 of them, including the epidermis. So just bear that in mind. That in the next, I would say, 2 minutes, complete the first column. The next 3 minutes after that, so 5 minutes in total. Those 2 columns should be done, the first 2. And if you have questions,
like right beside xylem. The circle beside the xylem is carbon green. That part. I think. Oh. Like you down. Yeah. So when the you have a good one. Oh, okay. The celery, okay, no, or wait, did you get the microwave? Oh, I have to do it like. Yeah. Okay, so that. That's pretty.
Yeah.
Just remember to take the photos, because you're going to use them for your presentation. And it's okay if you share them, but I just want to make sure that you're.
Where do you put the rest of the plant? You keep it or is it better?
Yeah.
Do we need the measurement for that?
I would say, just once again, to remember everyone, by now you should have completed the first two sections. And just as a hint, one of them is in present, and it's actually almost labeled and applies to the table itself. Right, so just think about that. One of the headings it says if present, so think about which of the ones that isn't, and that's something to help, but accordingly, you will have only 15 minutes to now officially complete the lab. So make sure that within those 5 cell types, each member uses at least one of the microscopes to measure within 3 minutes each cell type. And you just have to take the OMU. Then after at the end today, you can just multiply by the calibration factor. If it was at 10x, 25. If it's at 40x, it's 6. You can just bear that in mind, multiplying by that value. Then because of timing, we need to make sure that we're done in 15 minutes that round. So please bear that in mind. When you're done, TBO for cleanup, I'll talk about it in 15 minutes, but please try to complete as much as you can. Yes.
Like, probably, I think so. 300 seconds. I don't know about it.
Oh.
Oh. Yeah.
I use, I really.
Not much. Some, yeah. Oh, yeah. Rainbow. So I thought I was also beautiful. One point. 1.1. Yeah, I'd say anything above 1.0 is okay. Yeah. No, I don't think I do that. I used to pay a lot of oh, okay. Yeah. My feet's only nine. Like, if I say no, it's like this. No. Yeah. I kind of want to. I don't like the rest of it. It was just like a lot of variety. It's a lot of boxes. Nice. Oh. Oh, really? Oh, yeah. I've already had problems. The battery's terrible. Oh, yeah. Yeah. It's basically just like it's so so yeah, that doesn't work at all. They go to go. Okay. Oh yeah. Okay. Yeah. Okay. Yeah, I got to go with that. Yeah, yeah. Are we good to go? Or if you start like your other you ready? Oh. Yeah. If you could just speak about 3 minutes. It's one of these. Where did you get those from? No, that's okay. You finished that? Thank you. Oh. I. No, so I have MD42. Yeah. Yeah, I think you said the first two by seven. Yeah, I agree.
Well, everyone, this is your time to communicate now, at least, finalizing your results, because in the next few minutes, we actually have to begin cleaning up. So please make sure that you've already taken the second, the first two columns complete, right? As I mentioned earlier, there's one that's not included, but you have to make sure that you're able to at least get three, if not more, 3 to 5 values per cell type. And when your group is done, TBO is placed inside there where you can see them rinsing off the water off of the TBO plate. So bear in mind that you clean it up so that way future use can also be rooted. Also for your plants, you can take them out and place the containers inside there. Yeah. Some of you have already like cleaned up nearly all of your specimens. If you haven't, let me know.
Yeah, the first one was pretty good. Yeah. Okay.
So it's just gonna be like the abstract.
Yeah, I see one.
Citation. Other pronunciation kind of thing. I got a 19 tomorrow.
Okay.
I was worth 15%.
Oh, really? This one's only worth 5, I think, right?
Any other questions? I'm not going to
Yeah. I didn't see it. Yeah. So
Please let me know because I do want to finalize some post-lab reminders for you all. Just bear in mind if you can wait for about 4 minutes, that would be appreciated because it's important that everyone else is able to complete it. And if you can, help others that are trying to complete their portion of today's lab. That way, everyone can leave together and fulfill the rest of today's priority. I think some of you may have already anticipated on leaving. There's only a few other reminders that you're already ahead of the game, so just stay a bit more longer, and then you have to miss these five. Okay, so just bear that in mind. This one we Okay, now everyone, before you leave, and I just want to say I really appreciate your steadfastness, your patience. I don't want anyone feeling like intimidated and anxious at the last like 10, 15 minutes. And I know many of you probably are, but help one another if you can, so that way everyone in the lab can complete today's practical with ease. For those that are wondering, once again, 3 different cell plant types, right? Make sure that you're doing either epidermis, parenchyma, xylem, phloem. Those are your 5, right? So bear in mind that you're including all of the details asked for you: cell size, shape, location of the stem or relative to the stem, the cell wall thickness, the cell wall type, and even the functions of that plant or tissue cell type. Bear that in mind. And at the last, let's see. Yes, it's due at the last, like tomorrow. And then relate once again the functions, as you see here, for the arrangement of the tissue, the cell shape, the different parameters touched upon earlier. And make sure that you use the images that you collected from the light microscope. Hopefully everyone is almost done. With that, I really appreciate your time. All the best. Looking forward to seeing you 2 weeks from now. Take care. Bye. They could be thrown in the glass. Any remaining glass slides can be placed in the full black bin. Right inside. Any final questions? For those that don't have my email, it's my first name dot last name at utoronto.ca. Just want to make sure for those that came in and this being their first week, just to let you know, this is my name, SAID at utoronto.ca. So if you have questions, you can also reach out to the BioHelp email. They'll help you for your alpha Lars to prepare for your writing assignments as well. due on the 3rd of February. But with that, looking forward to seeing you in 2 weeks' time. And if you have questions, feel free to reach out. Thank you so much, everyone. Make sure that as well, hopefully everyone's trying to clean. Questions? Let's have a And don't worry, you guys, you guys are doing well in advance. Preparing to clean up 10 minutes in advance is better, so that way you don't have to rush yourself completing today's practical. And I understand maybe at times things may be tough, but just let me know. And I want to help you all succeed. So don't think of me being aiming to be tense in any way, shape, or form. So let's have a look. Some of you may be wondering, actually, I'll shut off with them and I'll just let you know. If it's not 2 weeks' time, it's going to be in a month's time, our next practical. I thought lab 4 would be after reading week, the first lab after reading week, but it's also in the manual. Let's see. We do have, yeah, so you will be coming back 2 weeks' time. Take care, you guys. If you have any questions, also let me know.
These look all right for values. These ones are 10x. So like 5 essentially millimeters per, and then these ones are 40. These ones I was looking at, they're like practically the same.
It's okay if they're, so for context, I want to be able to take the measurements, but if you have other features that are more correct, more accurate, I'll take those as well. So don't worry. If I also find that yours are accurate, then I'll make it even easier for you guys to get.
Great. Hopefully that answers your question. I don't know what that.
Everyone okay? How do we feel these last one? Don't worry, like, by the way, I don't want to give you my this because I don't have enough to prepare for our and you should do it. So that one thing the quickest alarm needed for review will be around 2 days. So we should have that time to be able to
I'm gonna doing good. If you actually use the other side, I'm good. Okay. By the way, do you think it's okay if I leave that there? Because I know sometimes they feel more helpful.